Okay, hi folks. Everybody okay out there? All right. So folks, can you hear me okay? All right. Okay, great. Hey, I apologize. I don't know what happened. My internet at my house went down for about 45 minutes.
It says that they have an outage and it may not be fully resolved until 10:45.
Okay, all right. So I don't know what the heck's going on here. So I'll make an attempt to continue with class, and if it goes out again, just stay patient. And we'll do the best we can. What lousy timing, because we got to get you ready for an exam. So it was curious to me. I don't know if anybody else had an internet issue, but I had an internet issue. It went dead on me. And I also was having a cell phone issue. I couldn't call anybody. I couldn't text anybody. I couldn't communicate to anybody in the world. And then it just started working later on, like 10, 12 minutes ago. So anyway, sorry about that. We'll do the best we can to make up ground here. I'm going to use some lab time to finish the conversation I need to have with you in chapter 10, because I need to get you ready for your exam. Okay, so bear with me. I'm gonna review what we were talking about and continue. Okay, hang in there, and let me know if you're having problems or if you have questions. Okay, let's continue. Okay, so we were talking about a neuromuscular junction and how the events at the neuromuscular junction are able to activate muscle contraction. So I won't go through nitty-gritty detail what I've done, but I would like to summarize so that we can get our minds up to speed again. Okay, so what I've shown you in the drawing is a neuromuscular junction. We have an axon here. We have a muscle cell. The green thing is the electrical signal known as an action potential. So what will happen is when your brain says to your arm, lift up, electrical signals will be sent down the axons that innervate your shoulder and arm muscles so that they contract. So this electrical signal spreads down an axon. The electrical signal is made of ions, right? Ions, charged atoms. And when those ions reach the proteins that we call voltage-gated calcium channels, that changes the voltage across the membrane, which is a fancy way of saying it changes the difference in electric charge between the inside and outside of the membrane. Long story short, the presence of the electrical signal, which is made of ions, changes the shape of the calcium channels, opens them so that calcium in the synaptic fluid of the synaptic cleft enters the synaptic end bulb of the neuron. Okay? Do you have any questions right now? Okay. All right, thanks for hanging in there. So we'll continue. Okay, we'll continue. What's going to happen next is that the calcium that has entered the synaptic end bulb, it is going to bind to special structures inside the neuron called synaptic vesicles. Okay, so let me get this drawing up here again. So inside the end bulb are these tiny little bubbles that are called synaptic vesicles. Okay, and I'm going to be running out of space here very soon. Again, you want to be looking in chapter 10 at the better drawing of your textbook than what I can give you, but it's figure 10.9. And so let me say that this is a synaptic vesicle. Okay, so the calcium will bind to the synaptic vesicle. And what will happen is that the synaptic vesicle will migrate. It will literally move by, the synaptic vesicle is a vesicle. Do you remember when we talked in chapter 3 about vesicular transport? So long story short, the synaptic vesicle will move to the inner surface of the plasma membrane of the neuron, fuse with it, and dump its cargo into the synaptic cleft. So I know this is kind of very disjointed at this point because we lost our internet service and things were disrupted, but let me ask you a question. If you're following along, you've been reading the chapter, let me see what you understand. Here we go. What substance is contained inside the synaptic vesicles? What's in there? What substance is contained inside the synaptic vesicle? What do you think, folks? Okay, so Christina says ACH, and that is a correct answer. How about the rest of you? Okay, I'm running out of colors here to use, but okay, I'll stick with purple here. So inside the synaptic vesicles is something called acetylcholine, and acetylcholine is a neurotransmitter, right? So when calcium enters the synaptic end knob, it binds to the synaptic vesicles and causes the synaptic vesicles to dump their acetylcholine cargo into the synaptic cleft, okay, by exocytosis. All right, so let's write some of these ideas down here. I'm going to just go below and then I'll come back up in the drawing. Okay, so in our notes, we had that calcium entered the neuron. The calcium ions bind to the synaptic vesicles. This triggers the release of acetylcholine from the neuron. And remember, acetylcholine is a neurotransmitter that's synthesized by neurons and stored in these vesicles. So the calcium enters the neuron, it binds to the synaptic vesicle, and it triggers exocytosis of acetylcholine. Okay, so we're doing pretty good here. I mean, in this piece of the conversation, we're almost ready to wrap up this part of our conversation. Okay, any questions? Okay, so acetylcholine has now been released from the neuron. Acetylcholine will float in the synaptic fluid, and it will diffuse across to the membrane of the muscle cell, the sarcolemma, where it will then, and again, forgive me, I'm not much of an artist here, but I'll use orange to represent something else here. So I'll put little triangles here in here, and we'll call these acetylcholine receptors. So the acetylcholine will diffuse across to the sarcolemma of the muscle cell and bind to the acetylcholine receptors. Okay, the acetylcholine receptors are just different proteins embedded in the membrane of the muscle cell, right? So they're going to bind here. All right, let me ask you let me ask you a question, and you'll be able to answer my question by looking carefully at figure 10.2, panel C as in Charlie, panel C as in Charlie. Here we go. Hold on 1 second, and here we go. Here we go. How many acetylcholine molecules bind to one acetylcholine receptor molecule to allow sodium ions to enter the cell? Take a look at panel C as in Charlie of figure 10.9 very carefully. Okay, thank you, Christina. How about the rest of you? I want to be sure that you're really getting this, that you're digging into it, you're looking at it, and you're understanding. So Christina's responded. How about the rest of you? Okay, good. Thanks so much, folks. And you know, for everybody that has responded, thank you, and everybody's responded correctly. So if you're not sure, the answer is 2. The answer is 2, and you can verify that by looking very carefully at picture C, as in Charlie, of figure 10.9. So 2 acetylcholine molecules will bind to each acetylcholine receptor. And when the acetylcholine molecules bind to the receptor protein, as you know, the receptor protein will change shape, because when any 2 molecules interact, they change the shape of each other. Okay, so the acetylcholine binds the acetylcholine receptor, and that causes that receptor to open a tiny little channel so that sodium ions, which are also floating around in the synaptic fluid. So I'll put a couple blue-colored sodium ions. These sodium ions will come down through the channel here, the channel that's formed by binding of acetylcholine to the acetylcholine receptor. Okay. All right, so number 4 in our notes, number 4. Okay. Acetylcholine, ACH, binds to the acetylcholine receptor. The receptor changes shape, and sodium ions, which are also present in the synaptic fluid, enter the muscle cell. Okay. All right. So obviously the drawing is getting very busy now, isn't it? So we risk getting too busy so that we lose understanding, and we need to be careful about this. But the acetylcholine is released from the neuron, it floats across to the receptor, binds to it, changes its shape, and sodium ions enter the muscle cell. What you may or may not be aware of at this point is that sodium ions are one of the ions that make up an electrical signal. So what I'm telling you is this: when the acetylcholine neurotransmitter binds to the muscle cell protein, that literally activates a brand new electrical signal in the muscle cell plasma membrane. Okay, so what's going to happen here, for lack of better drawing ability, is that the passage of sodium into the cell is going to start a muscle action potential. You notice up here in the neuron where I drew the neuron action potential as a jigsaw pattern of green going back and forth across the membrane of the neuron? Well, the sodium is going to start going back and forth It's not quite accurate, but that's what I'm going to tell you right now. Going back and forth across the membrane, the sarcolemma of the muscle cell. So entry of the sodium triggers the generation, the creation of a new electrical signal, not in the neuron, but actually in the muscle cell itself. Okay. Okay, sodium enters the muscle cell. This triggers a muscle cell action potential. Okay. All right. So here's now, any questions? Okay, let's continue. Now, you recall from our last conversation, last class, that we went over some of the anatomy of a muscle cell, and that was intentional because you need to know this anatomy before this conversation we're having now makes any sense. Do you remember that part of the muscle anatomy were these inward invaginations called T-tubules? Okay, so I'm not much of an artist, but I'm just going to draw some extensions into the interior of the muscle cell and call these extensions T-tubules. Okay, transverse tubules. So long story short, what's going to happen is as the sodium enters the muscle cell for the first time, you trigger a muscle cell action potential, which spreads along the plasma membrane of the muscle fiber. And since the T-tubules are part of that plasma membrane, that electrical signal is also going to go down and into the cell via the T-tubules. Okay? So you have this electrical signal that's really going deep into the interior of the anatomy of the muscle cell. Now, you also remember, I hope, from last time, that there was another membrane inside the muscle called a sarcoplasmic reticulum. All right, so I'm going to draw some other little fuzzy things and say, okay, here's another membrane. Okay, so this is the sarcoplasmic reticulum. Okay, so let me ask a question in the chat window to see if you're following. Here we go. What substance is stored in the sarcoplasmic reticulum? Okay, thank you, Sierra. I certainly want to encourage everybody to participate, but because of our time constraint, I'll just go with the first correct answer I see in the chat window. So thank you. It is calcium, right? So the sarcoplasmic reticulum contains calcium. And at least in my drawing here, I've used purple to represent calcium ions. So I'll stick with that convention, right? Inside the sarcoplasmic reticulum are calcium ions. And you can probably guess what's going to happen. When the muscle action potential, represented by this blue jigsaw pattern, goes into the T-tubules, it passes nearby the sarcoplasmic reticulum and changes the shape of special proteins embedded in the sarcoplasmic reticulum membrane, which act as gatekeepers for the calcium ions. Long story short, as the electrical signal spreads through the T-tubules, the channels in the sarcoplasmic reticulum open and calcium exits. It leaves the sarcoplasmic reticulum, and calcium is now floating around in the cytoplasm of the muscle cell. Okay, so let's continue with some notes here. Okay, number 5. Okay, so we talked about the action potential being triggered in the muscle cell. The action potential spreads into the muscle cell via the T tubules. As the electrical signal goes into the T tubules, it triggers the release of calcium from the sarcoplasmic reticulum. So we now have calcium floating in the cytoplasm, okay? So we're almost done with our conversation in this piece of the drawing of this process. I don't think I'd be capable of doing much more, to be honest with you, okay? So calcium is floating in the cytoplasm. And I'm going to ask a question in the chat window to see how well your study time is going. Hold on a sec. Here we go. What protein does calcium inside the muscle cell bind to allow muscle contraction to proceed? Okay, let me give you a hint. Here's my hint. The answer to the question can be identified by looking very carefully at figure 10.7, panel B as in boy. Figure 10.7, panel B as in boy. Okay, what protein does calcium bind to? Okay, great. Christian came up with the answer correctly. So I'll continue and everybody try to locate that. The answer is troponin, right? Troponin is one of those regulatory proteins we put in your notes last time. So long story short, the calcium that's released from the sarcoplasmic reticulum floats around and binds to troponin protein. When it binds to troponin protein, this begins to activate the physical process of muscle contraction that we need to talk about next. Okay, so calcium binds to troponin to activate muscle contraction. Okay. All right. Folks, I'm going to step you through this entire flow sheet one time quickly and ask you if you have any questions afterward. Okay. So up here in our cryptic drawing, the whole process of muscle contraction starts by having the nervous system agitate a muscle cell. So this will begin when a neuron, represented by this blue bulby thing here, the neuron generates its own electrical signal made of ions. The ions go back and forth across the membrane of the neuron until you reach the end of the axon. Some of those ions will bind to the voltage-gated calcium channel protein, change its shape, allowing calcium ions that are stored in the synaptic cleft to enter the neuron. The calcium ions bind to the synaptic vesicles, which contain the neurotransmitter. When calcium binds to the vesicle, the cell knows to perform exocytosis to dump the neurotransmitter, in this case, acetylcholine, into the synaptic cleft. The acetylcholine moves across to the membrane of the muscle cell by simple diffusion. Two molecules of acetylcholine bind to the receptor, changes its shape, and when it changes its shape, it creates a tiny little channel for sodium ions, also present in the synaptic cleft, to enter into the muscle cell. That process is like a trigger that activates the creation of a new electrical signal in the muscle cell. So you get this new muscle action potential spreading along the sarcolemma and down into the T tubules, which are extensions of the sarcolemma. As that electrical signal spreads into the T tubules, it promotes the release of calcium from the sarcoplasmic reticulum. The calcium then goes over and binds to troponin protein, which I've highlighted in your notes, but I have not drawn in your drawing here in the notes, but it is in figure 10.7. Once you bind calcium to troponin, it's like knocking down a domino, because once you knock down the first domino, other things are going to happen, right? Boom, 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 boom. Binding of calcium to troponin is knocking down the first domino on our way to actually contracting a muscle fiber. Folks, do you have any questions about this process? I will ask you again on Thursday if you have any questions for the exam, right? So maybe you want to think about this a little bit. And let me say this, I've given you an awful lot of detail about this process, but I will also admit to something. I have not told you everything I'm going to ask you on the exam. Everything I'm going to ask you on the exam requires you to think, to reason, to logically deduce, and it requires you to really read your textbook, okay? So that's a challenge to you that I want you to take seriously. All right. If you don't have any questions about this, we'll continue. So the next thing that I can say to you is, trying to get another page in here. Okay, so now I want to, there we go, good. Something actually worked with the technology. Okay, good. Okay, folks, so now we have a description of events at the neuromuscular junction. Now we need to describe, or at least cover some basic ideas about the actual physiology of muscle contraction. And for this, we'll be talking about the sliding filament. Some textbooks refer to it as the sliding filament mechanism. Others refer to it as a sliding filament model. Looks like your book is using mechanism, so I'll mention that sliding filament mechanism, which includes the contraction cycle of figure 10.6. And I will highlight again that I'm going to show you another Wiley Plus video, okay, for under the 3D physiology. And this one is going to be, this one's going to be muscle contraction and the sliding filament mechanism. Okay, muscle contraction and the sliding filament mechanism. There are certainly additional videos on YouTube. If you typed in sliding filament model or sliding filament mechanism or, muscle contraction into YouTube, you would find some other videos, but you've already paid a lot for your course material. So why not use some of these videos, which honestly are pretty good quality. Okay. All right. So this video, I think, is about 5 or 6 minutes. So let's get it started so we can talk about it. Here we go, folks. Thanks for your hanging in there. I know it's been a difficult morning. Here we go.
An action potential propagates along a single motor neuron from the brain or spinal cord to the end of the motor neuron near the skeletal muscle cell. This triggers the events at the neuromuscular junction that result in an action potential arising and propagating along the muscle cell membrane or sarcolemma. As the impulse moves down the sarcolemma, it continues to travel deeper into the muscle cells through membranous structures called transverse tubules. The interior of the skeletal muscle cell is filled with rod-like structures called myofibrils. Each myofibril is formed by repeating structures called sarcomeres, which are the muscle cell's contracting unit. Each myofibril is covered by smooth endoplasmic reticulum, called the sarcoplasmic reticulum, which stores calcium ions. Sections of sarcoplasmic reticulum are located along either side of the transverse tubules. As the action potential travels down the transverse tubules, voltage-dependent calcium channels in the sarcoplasmic reticulum open and release calcium into the sarcomeres. The calcium activates the sarcomere to contract. Each sarcomere is made up of 3 types of filaments: tighten filaments and thin and thick filaments. The tighten filament acts to provide elasticity for the sarcomere. Thick filaments are surrounded on each end by 6 thin filaments. When the sarcomere contracts, the thin filaments slide over the thick filaments by a process known as the sliding filament theory. Thick filaments are composed of 2 myosin filaments twisted around each other, and which are anchored in the center of the sarcomere. Each myosin has a head and tail. There are binding sites for ATP and for actin on the head of the filament. Energy, in the form of ATP, moves the head into an activated position, much like cocking a gun. Once bound to actin in what is known as a cross bridge, the myosin head will bend and move the actin. The thin filament anchored to the ends of the sarcomere is primarily composed of actin proteins. Each actin protein has a binding site for myosin. The binding sites on actin proteins are covered by tropomyosin, which is maintained in place by troponin. Troponin is a protein spaced along the thin filament and attached to both actin and tropomyosin. It also has a binding site for calcium ions. In the absence of calcium, the myosin filaments are prevented from binding to actin filaments because the binding site on actin for myosin is covered by tropomyosin. However, when the sarcoplasmic reticulum is stimulated, it releases calcium into the sarcomeres. The calcium ions then bind to troponin. Troponin then changes shape and pulls the tropomyosin to the side, allowing myosin to attach to actin. When myosin forms a cross bridge with actin, then the myosin head bends as it slides actin toward the center of the sarcomere. Contraction of the sarcomere is a series of stepwise cycles occurring between the thin and thick filaments simultaneously throughout the sarcomere. The first step of the contraction cycle occurs when ATP, already bound to the myosin head, is hydrolyzed. This provides energy to position the myosin head in its cocked position. Next, when calcium ions are released from the sarcoplasmic reticulum and attached to troponin, it uncovers the binding sites on actin. Myosin forms a cross bridge with actin. Once this occurs, the myosin head bends from its cocked position and slides the thin filament towards the center of the sarcomere. This process is referred to as the power stroke. In the final step for each contraction cycle, ATP binds the myosin head, resulting in detachment of cross bridges. As long as the levels of calcium and ATP inside the cell are adequate, then the contraction cycles will repeatedly occur and the sarcomere will shorten. That is, as long as the skeletal muscle contains enough energy and is stimulated by nervous impulses, a continual shortening of the sarcomere will occur. Calcium pumps in the sarcoplasmic reticulum pull the calcium ions out of the sarcomeres back into storage. When the calcium ions are removed from troponin, it returns to its original configuration, allowing tropomyosin to again cover the binding sites on actin. Myosin can no longer attach to actin, and the tightened filaments pull the thin filaments outward to their original relaxed position. Due to many contraction cycles, the actin and myosin filaments will slide along one another to shorten the sarcomere. When no longer stimulated, the sarcomeres will return to their previous length.
Okay, folks, so there you have it in a nutshell. And certainly that presents the information in a graphical, cartoony way better than I could potentially draw it. But I do intend to walk you through some things in your notes. Do you have any questions about this process from having watched the video one time? Okay, all right. So please let me know along the way if I confuse you, okay, if you have questions. We're going to try to finish this up in 20 minutes so that we can get into our lab content today. Don't forget, if you go to study this content from the recorded video of today's class, there's actually going to be 2 videos from today's class, because when the internet went out, my computer immediately started building the video because the meeting had stopped, and I'm also recording the lecture now. So you actually have 2 recordings for today's lecture. Okay, so first let's talk about the regulation. Let's talk about turning muscle contraction on and off. Okay, and I'm not going to draw here because I'm just not that capable of doing it. But, you know, we're going to start with this idea from our last notes where calcium binds to troponin. I will remind you that there are 2 important proteins that regulate the contraction process. And by regulate, I mean control whether contraction can happen or not happen. And that's troponin and tropomyosin, okay? So when muscles are relaxed, meaning there is no muscle contraction, the muscle contraction itself is kept suppressed. It is kept turned off because tropomyosin, which the video highlighted and shared with you, the tropomyosin protein is blocking certain binding sites on actin that myosin wants to grab, okay? You can probably see this best on figure 10.4. Figure 10.4. Okay. And so I'll reference this as a way to think about it when you're studying. When muscles are relaxed, meaning there's no muscle contraction, tropomyosin and troponin are positioned in such a way that myosin cannot bind to actin. Okay? Tropomyosin and troponin are positioned in such a way that myosin cannot bind to actin. We need myosin to bind to actin if we're going to begin to shorten a muscle fiber. And if that can't happen, then you can't contract. Okay? So if the nervous system wants your muscle to contract, it will do the series of events we just drew out over the past 30, 45 minutes of lecture. It will do these events, release acetylcholine, which will stimulate the muscle fiber to release calcium from the sarcoplasmic reticulum. The calcium goes and it binds to troponin. Okay. The nervous system wants a muscle to contract. Okay. The nervous system triggers through a whole bunch of events I'm not going to describe again, right? Because we just described them. The nervous system triggers the release of calcium from the sarcoplasmic reticulum. Okay. The calcium will bind to troponin. When I say calcium, it's calcium ions. Okay, Ca+2. So I'll just put it this way. Ca+2 binds to troponin. Let me ask you a question in the chat window to make sure you're following along. Here it comes. When calcium binds to troponin, what characteristic of troponin changes? When calcium binds to troponin, how will troponin change? What characteristic of troponin will change? It's a common biological principle that we're talking about repeatedly this semester. When calcium binds to troponin, what characteristic of the protein will change? Sierra says the shape, and Sierra is absolutely right. And I hope the rest of you understand that, okay? When calcium binds to troponin, troponin changes shape. When troponin changes shape, because troponin and tropomyosin are touching each other, I mean, as you look at the pictures in figure 10.4 and figure 10.6, you'll notice that troponin and tropomyosin are touching each other. So when troponin changes shape, that promotes a positional change in tropomyosin, so that tropomyosin, if my top hand is tropomyosin, when troponin binds, when calcium binds to troponin, troponin changes shape. That causes tropomyosin to rotate its position along the actin. That exposes the actin binding sites that myosin wants to grab onto. So troponin changes shape, which prompts a change of position of tropomyosin. Okay, tropomyosin changes its position. This exposes the binding sites on actin that myosin would like to attach to. Okay, if myosin has the ability to bind to actin, we can contract the muscle fiber. But if myosin cannot bind to actin, we cannot contract the muscle fiber. So the whole ability to contract depends on exposing or blocking the binding site on actin that myosin wants. And that's regulated through the troponin tropomyosin complex. All right, so calcium binds to troponin. Troponin changes shape, which prompts a change of position of tropomyosin. This exposes the binding sites on actin that myosin would like to attach to, and contraction can occur. Now, we haven't told you the steps of the contraction. We're going to do that next. But what we're describing right now is the regulation of the contraction. Okay. All right. You might say, well, okay, your muscles are contracting, but how does the muscle contraction turn off? Well, the muscle contraction will naturally turn off the muscle contraction will turn off whenever your brain says, I don't want to contract anymore. And what that means is when your brain says, I don't want to lift my arm up anymore, so I don't want to lift it up. When that happens, the neurons of your brain stop sending electrical signals to the muscle fibers, which stops promoting the release of acetylcholine, which stops promoting the release of calcium from the sarcoplasmic reticulum inside the muscle cell. So we stop electrically exciting the muscle fiber. And if we stop electrically exciting the muscle fiber, it will go back to its resting position naturally. And how do you do that? Well, what will happen is, you saw in the video that the sarcoplasmic reticulum contains tiny little calcium ion pumps. And you can imagine like you're sucking on a straw to drink some milk, right? Okay, imagine that kind of activity happening inside the cell for tiny little straws, tiny little pumps embedded in the sarcoplasmic reticulum that are going to gently draw calcium back into the sarcoplasmic reticulum. Okay. When the nervous system stops electrically exciting the muscle fiber, calcium ions will be pumped back into the sarcoplasmic reticulum. Okay, so let's think about that. If the calcium ions that a second ago were attached to the troponin are being sucked off by the pumps back into the sarcoplasmic reticulum, if they're being detached from the pumps, then what that means is troponin is going to go back to its original resting shape, which will allow tropomyosin to shift back across the actin, blocking the binding sites that myosin would normally be attached to. Okay, so removal of calcium from troponin causes the troponin / tropomyosin complex to reposition itself and block the binding sites on actin that myosin needs to contract. Okay, so this is how you shut the muscle contraction back off. Okay. All right, folks, any questions? Okay, so we've just covered how you turn the muscle contraction on and off. Now we need to at least step through a couple ideas about, well, when the muscle contraction is on, what actually happens at the molecular level to allow the muscle fiber to shorten. So number 3 here, okay, and we will finish our conversation in this chapter with this, is the contraction cycle. Okay, the contraction cycle is figure 10.6. The contraction cycle attempts to explain our, and by our I mean science's best understanding of what happens here. It's not perfect. There's still some mysteries to this process that science hasn't fully figured out yet, okay? But is our best understanding of the physiological processes happening when muscles actually shorten. Okay, the events that occur during muscle cell shortening. And I'll stick with my labeling here, but I'm going to reference the information in figure 10.6. Okay, so think about it. This is the contraction cycle. A cycle implies a circle. And you have to ask yourself, is there a beginning or an end in a circle? Not really, right? A circle is 360 degrees. I could arbitrarily define start and end at any location of the circle because it's a circle. Okay, so the contraction cycle is something that starts, occurs, and repeats. Starts, occurs, ends, repeats. Starts, occurs, ends, repeats over and over and over again until the nervous system stops electrically exciting the muscle fiber. So we'll use the terminology and vocab and imagery of figure 10.6 here. Okay. So in figure 10.6, you see different color-coded balls there, right? Number 1, 2, 3, and 4. Number 1's at the top of figure 10.6. So we have blue ball 1 here. And so what we're saying is that in this cyclical process, the myosin head that wants to be part of the contraction, the myosin head is going to break down a molecule of ATP that allows the myosin head to position itself properly for step 2 when the myosin head actually grabs onto the actin. Okay, so myosin breaks down ATP to release energy that can be used to position the myosin head near the actin protein. So in the next split second of time, myosin is able to grab onto the actin. That's going to be step 2, what we're getting ahead of ourselves here. So myosin breaks down ATP to release energy that is used to position the myosin head near the actin protein. Okay. Question in the chat window for you. Coming up. Here it comes. When myosin breaks down ATP, myosin is behaving like N blank. Fill in the blank for me, folks. When myosin breaks down ATP, myosin is behaving like N blank. So here's what I mean by on the exam, you're going to have to think about things. It's not all about regurgitation. If myosin is breaking something down, myosin is acting like N what? I know you can do this. Come on. Okay, I'm going to give you a little hint. Okay, little hint. Hold on. Here we go. Here's my hint. The answer starts with the letter E. Okay, Mark, well done. That's exactly what I'm fishing for. It's an enzyme. So folks, what I'm telling you is that myosin, which we've defined for you in the notes last time, it's a protein. It's a structural protein. It is a structural component of the sarcomere, which itself is a section of a myofibril. The myosin protein is a structural protein. It forms the structure of the contractile machinery of the muscle cell. But even though I can describe it as a structural protein, I can also describe it as an enzyme because it is responsible for catalyzing the breakdown of ATP. It's just like I can describe you as my student, but somebody else might describe you as their son or daughter, or someone might describe you as their mom or dad, or they might describe you as an employee or a volunteer. You're the same person, you can be labeled different ways. Molecules are like that. Myosin is a structural protein. It also happens to behave like an enzyme, so it's an enzyme. It's both things at the same time. Okay, so next, blue ball 2. Okay, blue ball 2 in figure 10.6. You know, the energy released from breaking down ATP in the first step, blue ball 1, is going to be used for myosin to literally grab on to actin, right? Going to grab it, going to bind it, okay? So when you do that, you form what's known as a cross bridge. When myosin is attached to actin, you have formed a cross bridge. The energy released from blue ball 1 above is used by myosin to grab onto actin, okay, forming a cross bridge. Blue ball 3, okay? So what will happen here, just take a look in your textbook, the myosin head is going to pivot, which is a way of saying the myosin head is going to move. It's going to move, and as it does so, it's going to drag actin along for the ride, because at blue ball 2 there, at step 2, myosin just grabbed actin, right? So if my left hand is myosin and I grab my right hand, which is actin, if I move my left hand, my right hand goes along for the ride, right? That's what's going to happen here. So myosin moves towards the center of the sarcomere. In step 3, I very much like the language of your book because it says the myosin head pivots, pulling the thin filament made of actin past the thick filament made of myosin toward the center of the sarcomere. Myosin pulls actin toward the center of the sarcomere. And you can appreciate if my hands represent the distance between the ends of a sarcomere, if something's moving towards the end of the sarcomere, if something's moving towards the center of the sarcomere, we're going to shorten the sarcomere. We shorten the sarcomere, we're going to shorten the myofibril, and we're going to go through that pathway we started the class with this morning, right? Okay. Myosin pulls toward the center of the sarcomere, the muscle fiber begins to shorten, meaning contract. Okay. Now, obviously, we're talking about molecules. They can only move things a little distance. So if I talk about my biceps brachii contracting to pull my hand towards my face, if I just have a couple myosins contracting, right, I will move my arm just a little bit. You can barely notice it. So if I want to complete the entire contraction, I've got to repeat this process over and over and over again, kind of like I like to think of the motion of myosin as oars in the water in a rowboat. Think about a row team in a rowboat where the oar is the myosin, right? We pull the ore out of the water, push it forward, push it down into the water, and pull. We shorten. We pull the ore up, we push it forward, we slam it back into the water, and we pull again to go further with the boat, which is to shorten the muscle fiber even more. Okay. So what's going to happen here? You notice between step 3 and 4 in your book, notice that ADP is released from the myosin. And so for step 4 here, you will have a new molecule of ATP, attached to myosin. When it does so, the attachment of the new molecule of ATP causes myosin to change its shape, release its grip on actin, and fall away. That's like the ore coming out of the water and pushing forward so I can begin another contraction process, okay? So a new molecule of ATP binds to myosin, causing myosin to momentarily let go of actin so that at step 1, right, blue ball 1, I can begin the entire process over to continue muscle contraction. Okay, think about the rowboat analogy with oars. Okay. All right, folks. So I know that's probably more than you've ever wanted to know about muscle contraction physiology, but this is the business we're in right here. Okay, this is what we're doing. Right. So when we have problems with muscle contraction, there's lots of questions you could ask about this. I mean, people often ask me about, muscle spasms and things like that. Well, muscle spasms can be caused by a lot of different problems in the body. They can be caused by a problem with the nervous system sporadically activating the muscles to contract. You can also have a problem with the calcium inside the muscle cell where you get inappropriate release of calcium from the sarcoplasmic reticulum, even when the nervous system wasn't triggering that release, and that can cause, muscle spasms and things like that. Some of those things can happen because of injury or, kind of bruising and damaging injuries to the muscle. If you've been playing football and somebody rammed their helmet into your arm or leg muscles, that can bruise the muscle, it can damage it and promote, spasms and things like this to happen with the muscle tissue. All right, folks, I think I've basically covered what I can reasonably cover here with you today. Do you have any questions you'd like to ask about the neuromuscular junction or regulation, turning on and off muscle contraction, or the actual shortening of the muscle fiber that we just talked about in relation to the contraction cycle? Anything I can try to clear up for you? Okay, folks, then I'm going to stop recording my lecture since we're done with this part of our time together. Okay, and again,